All right, it's 5:01, Liz. Does it look like
All right, let's do it.
People are in? Okay. Hello and welcome to the first event. Oh, no video, okay. The first event of the West Maui Ridge Reef Rendez-VIEW for 2020. As many of you know, this is an event we've held annually for the last 10 years as an opportunity to get our partners together from Ridge to Reef, to share in all the wonderful things that are taking place and give a chance to connect with the good work, with the volunteer community, and with the key messages that are taking place. And I'd be lying if I didn't say I was sad that I'm not going to see you all at some point during this month, but I mean, how wonderful. We had nearly 150 people sign up for this talk. So thank you for making the migration to the virtual Rendez-VIEW format for this year. And I'll just take a minute to plug the other things we have lined up for this month. So I hope you've had a chance to go to the westmauikumuvai.org site. I don't know if Liz, you're going to throw that up there.
Yes.
We are going to have lots of messaging from our conservation partners, so you have a good excuse to be on your Facebook this month, where we're featuring our various partner groups for a couple days at a time, and good things coming from all of them. You can sign up for a virtual scavenger hunt. It's time to get your quiz night team together. I've heard some bragging taking place already. So quiz night will be at the end of the month, at the 30th. So get your team together and win some prizes on Ridge to Reef and pop culture related questions that we'll be having there. And of course, our science night series. So every week this month, today's Tuesday, but from here on out Wednesdays, we're bringing some interesting speakers for you to learn from and engage with, and we have that lined up for tonight. But before we get there, I'm just gonna bump it over to Liz for a second, who's going to explain the technical part of how we're running this.
Thanks, Tova. Hey, everyone. It's nice to see so many of you. Like Tova said, it's awesome that so many people are interested in signing up for this, and we miss all of you in person. So yeah, I'm gonna be the tech person, which is, I haven't done this before for a webinar like this, so bear with me. If you have any questions, if there's issues that you're seeing, you can type a message in the chat. I think you can even make it private to me. I'm continuing to admit people. We are recording this, so we're hoping to put it on YouTube, and I'm seeing if I can also live stream to Facebook at the same time. We don't know yet. Now, at the end, we'll have some time. So if you have questions, feel free to post them in the chat. Just type them in there if you see the chat box. And if you're addressing any one of our three speakers in particular, note that as well. And we should be able to have some time at the end for questions. Yeah, so if you or someone you know who wanted to be part of this wasn't able to tonight, we will send out the link later, assuming all the recording works. And I'll try to get us streaming onto Facebook right now and continue to admit people who are joining us. So thank you again, everyone. And I'll throw it back to you, Tova, to do the proper introductions.
Thanks, Liz. Those of you who know the dynamic between me and Liz, we're a great team, but nothing virtual would happen without Liz because I'm totally useless in the online space. So before we get into our talk, I just want to take a minute to introduce our wonderful speakers. So Yina Liu is an assistant professor of oceanography at Texas A&M University. And if when you saw her picture, you were as curious as I was, she's wearing a parka because she was actually in Antarctica sampling for dissolved gases in seawater. And she was looking at these because they relate to the catalytic destruction of the ozone layer. Yeah, I thought we had that one under control when I was a kid, but apparently not. She is really interested in understanding how these organic compounds are impacting the planet and its functioning. And this is why she's joined up with the team here to contribute her knowledge to work on this project. Thank you for being here, Yina. Katie Shamberger is a California girl. She grew up in Huntington Beach. And being in California, she always wanted to study the ocean. Lucky her, after a semester abroad in the Caribbean, she fell in love with reefs and been studying them ever since. But she's no stranger to Hawaii. She did do her master's here at UH Manoa before then doing her PhD at the University of Washington in Seattle, both of which were in chemical oceanography. And you might be wondering, what's the connection between these three ladies? Well, she was super lucky to have Andrea as her very first PhD student. So like Yina, she's currently an assistant professor at Texas A&M in the Department of Oceanography. Which brings us to our very own Andrea Kealoha, who was born and raised on Maui here in Paia and has brought her talents back home to the University of Hawaii Maui College, where she's heading up the establishment of the first water quality testing lab on the island. Which, if you don't know about this, it's incredibly exciting because it's going to raise the capacity for us to understand our water quality issues on island without having to ship things off. So super grateful that she came back and is here helping the islands do better at looking into this kind of issue. So I will now bump it off to you ladies. Thanks so much for taking the time and joining us. Some of whom are in time zones that make this very late, so be patient.
Oh, there we go. Okay, can everybody see that? All right, well, thanks Tova for the introduction. We are really excited to give this talk. We're excited about this research. So thanks to all of you for signing on this evening. And so our research is looking at how water quality and coral reef health are responding to the population reduction as a result of COVID, and subsequently a wastewater reduction and injected wastewater at Kahekili. So coral reefs are important to people, to ecosystems, to economies. They're one of the most biologically diverse ecosystems on the planet, right up there with tropical rainforests. And so many ocean organisms depend on healthy coral reefs. They provide food for hundreds of millions of people around the world, particularly in developing countries and in places like Hawaii, where we're surrounded completely by the ocean. They provide coastal protection. So coral reefs dissipate a lot of the incoming wave energy that would otherwise cause more erosion to our shorelines and to shoreline structures. And then from an economic perspective, the value of coral reefs is estimated at tens of billions of dollars per year. And in Hawaii, we really rely on healthy coral reefs for our tourism industry. So there's a number of different stressors to coral reefs from human activities and from climate change. And we're kind of gonna focus on the ones that are most specific to Kahekili. So Kahekili is located on the west side of Maui, right past Kaanapali. So the Lahaina Wastewater Reclamation Facility receives wastewater from the Lahaina Kaanapali area, treats that water, and then injects it into injection wells. And that water then mixes with groundwater, flows towards the coast, and then is released in seafloor springs at Kahekili. So this water is treated, but it still has high nutrient concentrations, low pH, so it's acidified, and potentially has high concentrations of emerging organic pollutants. And these all have the potential to negatively impact coral reef ecosystem health. So if you were to go out to Kahekili and snorkel around, you would notice that the corals located further away from the seeps are healthy, vibrant, colorful, and then as you move progressively towards the seeps, you're moving towards an ecosystem that is more dominated by algae. So corals like to live in nutrient-low environments, that just the conditions that they've adapted to living in. And so when you increase nutrient concentrations, you can create a whole lot of issues for coral reefs. The first is decreased water quality. So simply by just changing the natural chemical composition of the water that corals like to live in. High nutrient concentrations can lead to algae blooms, which has been repeatedly observed at Kahekili. And then corals and algae kind of tend to compete for space on the reef. And so nutrient enrichment can create an environment that is more favorable to algae growth. And so the ecosystem can shift from one that is more dominated by corals to one that is more dominated by algae. And then there have been a number of studies showing that nutrient enrichment, including Nancy Prouty's work at Kahekili, showing that nutrient enrichment and low pH water increases bioerosion rates. So this directly affects the structure, the skeletal structure of corals, and can increase susceptibility to physical and chemical breakdown.
Okay, I think it's my turn to talk about another kind of a stressor for coral reef, which is the emerging organic pollutants. The reason for looking at emerging means they are not the kind of the legacy contaminants that we're traditionally looking at. For example, things coming from petroleum and older generation of firefighting foams. Emerging are the new kind of contaminants that we start to identify that they're a threat to both human and ecosystem health. So the two groups of compounds that I'm looking at, working with Andrea and Katie, are the per and polyfluoroalkyl substance, PFAS, which is coming from mostly firefighting foam, the different formulations in the new generations, as well as the non-stick cookware, or any water repellent substance. For example, unfortunately, the waxy paper or any water repellent texture of the fast food packagings, pizza box and things like that, they actually do contain various concentration and types of PFAS. So I should also note that PFAS is actually a group of compounds instead of just one. Now we have more than 5,000 known entities of this kind of compound. So they're water soluble, so they move around in the ecosystem quite readily. And the other group of compounds that I'm interested in this project is the pharmaceutical and personal care products, including basically sunscreens and some painkillers, or even caffeine that are human use quite readily. So Andrea, can you advance one slide, please? So many personal care product, or PPCPs for short, are known to be endocrine disruptors that actually include ibuprofen, known to be a kind of a painkiller, and naproxen, etc. And these kind of compounds could end up in surface water or even marine water through the graphic that is shown on the right-hand side of the slide. So they could actually come from wastewater, sewage, going through the wastewater treatment plant. Unfortunately, many current technology actually are not designed to remove this kind of compounds from the water stream. So some of them do end up in surface water and later potentially in the marine ecosystem. And sunscreen, of course, they can come from directly whoever that swim in the water apply sunscreen in the water rather than going through the whole cycles of sewage or water treatment plant. And so these kind of compounds are known to pose a threat to ecosystem and humans as well. But some compounds that seemingly actually okay for humans, for example, the artificial sweeteners and caffeine, can also be an indicator of wastewater tracer. So this will be another group of compound that we can look at and try to trace what kind of a water source that might have end up in the ocean in this study. Andrea, can you advance one slide, please? And now going back to PFAS, if you guys are reading some news, you may actually see people start to call this kind of compound forever compounds because they do not break down easily in the environment. This poses a huge challenge in the treatment of this kind of compounds. And on the left-hand side, you can see graphics. They actually can come from water repellent raincoats, fast food packaging, as I just mentioned, and non-stick cookware. Unlike many traditional contaminants, they are actually water soluble. That means once they are applied as a firefighting foam in ground or being used in household substance and end up in the wastewater stream, they do finally end up in landfills and subsequently maybe in groundwater or any surface water pathway. So these kind of compounds, PFAS, are known to pose a significant human health threat. For example, it can cause various kind of cancer. It is also known to be endocrine disruption and also affect reproduction processes and also weaken immune systems. So by looking at this kind of compound, it not only helps us to understand how they move around in the watershed, but how much they do end up in the coral reef system will allow us to understand how this kind of emerging contaminants may pose threat to a very vulnerable ecosystem on the planet. I think that's my portion. Yeah.
Okay, so I'll talk about acidification. You've maybe heard the term ocean acidification, and usually when you hear the term ocean acidification, we're referring to ocean acidification that's driven by carbon dioxide or CO2 that's coming from the atmosphere. And Andrea and I do a lot of work on this. So humans are producing lots of carbon dioxide from burning fossil fuels, oil, gas, coal to produce energy. This carbon dioxide goes into the atmosphere and then dissolves in the ocean, where it reacts with water to produce an acid called carbonic acid. That's the one in the middle, the H2CO3. Because it's an acid, it releases a hydrogen ion, which is the pink H+. So that is the term ocean acidification. When you release that pink H+, it lowers the pH and causes the water to become more acidified. But at Kahekili, what is happening is that the submarine groundwater discharge also has low pH. So the submarine groundwater that's being released into the reef system is also adding these pink hydrogen ions into the water and lowering the pH. And this is another sort of form of acidification that we refer to as coastal acidification. So anytime we have pollution from land that is also causing acidification to the ocean, we refer to that as coastal acidification. So this is not a unique process that's happening at Kahekili, but Kahekili is a great example of coastal acidification. And the reason why we're really concerned about acidification when it comes to coral reefs, whether it's ocean acidification from the atmosphere or coastal acidification, is because those pink hydrogen ions react with carbonate ions, and that's shown here in yellow. And the reason why we care about carbonate ions is because the reef structure, that hard, three-dimensional complex reef structure that provides habitat for these incredibly diverse ecosystems, is made of calcium carbonate, this CaCO3. And you need calcium and carbonate ions to make calcium carbonate. So when those pink hydrogen ions that are released by acidification react with carbonate ions, they're basically using up the carbonate that the corals need to make the reef. So whatever kind of acidification is happening, it's leaving less carbonate ions behind. It's making it more difficult to produce that calcium carbonate reef. And corals are the most important organism on a coral reef that's making the calcium carbonate, but they're certainly not the only. There's lots of other important calcifiers. So acidification decreases the amount of calcium carbonate you're making, and it also increases erosion because it makes the calcium carbonate on the reef weaker. And it also increases dissolution. So some of the calcium carbonate can actually dissolve on the reef. And both of these things contribute to the breakdown of the reef structure. And we need that three-dimensional reef structure to support the coral reef ecosystem. So As acidification continues, what you get is a transition from this healthy, three-dimensional, diverse reef ecosystem to a low-lying, less complex, less diverse ecosystem that doesn't provide the same sort of services and doesn't support the same type of ecosystem that we like to think of as a healthy coral reef. So we're looking at changes in water quality, 3 different types of water quality: nutrients, emerging organic pollutants, and acidification. And we want to connect these to changes in coral reef health. And there's a lot of different ways that we can look at coral reef health. One of the most common ways is to do surveys of who is living on the reef. So you can do surveys of coral, surveys of fish, surveys of algae, surveys of all sorts of different organisms that are living on the reef. And this is really important for telling you who is there. And that's important for telling you something about the health of the ecosystem. Although you're attending a talk that is being given by 3 different chemists. So we're not doing that sort of important ecological work, although we collaborate with ecologists that do this important work. So combining our work with their work is really important. You can also look at the health of individual organisms. So you can collect corals or fish or crabs or any sort of organism that you're interested in, put them in tanks and get more detailed information on their health. You can even look at genetics, which give you really detailed information on the health of individual organisms. But the work that we're doing is looking at the health of the reef ecosystem as a whole. And we're doing that by looking at coral reef metabolism. And so what do I mean by coral reef metabolism? So I Googled the word metabolism, and the Mayo Clinic defines it as the process by which your body converts what you eat and drink into energy, which is probably what you think of when you think of the word metabolism. We eat food, your body turns that into energy to do all the things your body needs to do. We need to breathe, we need to move, all the things we need to do requires energy, and we get that energy from food. And that's the process of metabolism. So where do coral reefs get their food, and what do they need to do that we're interested in? So coral reef metabolism, again, we're using as a measure of the health of the coral reef ecosystem as a whole. So coral reefs also need food, just like we do. They consume food, all sorts of different types of food, small particles, larger particles. Some of this food comes from the ocean and gets transported onto the reef from afar. Coral reefs are also known for being very efficient recyclers. So there's food being produced on the reef, food being eaten on the reef, and it's being recycled within the reef ecosystem. So when that food is used up and turned into energy, that's called respiration. So we do the same thing. We eat food, we respire it to make energy, and we breathe out CO2 when we're respiring. A reef does the same thing. So respiration is one of the metabolism processes that we're very interested in for coral reefs. But reefs also get a lot of their energy from photosynthesis. Algae are a critical part of the reef ecosystem. Corals have symbiotic algae that live within their tissues and provide a lot of the energy that the coral needs to grow and reproduce. And there's also other important algae on the reef that are photosynthesizing as well. So that's another part of coral reef metabolism that we're interested in. Another critical function that a coral reef does that we are very interested in is calcification, right? We want to know that the reef is growing, making that calcium carbonate to support the three-dimensional reef structure. So we're interested in how the reef is growing. And then we also want to know about dissolution. So is the coral growing? Is it making that calcium carbonate? And is it dissolving? Even on a healthy reef, there are natural times when that calcium carbonate structure gets dissolved, especially by organisms that are living on the reef. Clams, worms can bore into the reef and dissolve some of that calcium carbonate. So how in the heck do we measure this? And it turns out you can do that using chemistry. I don't know if any of you ever put an exclamation point after the word chemistry. That's what happens when you're a chemist. It gets very exciting. So when we breathe out carbon dioxide and we breathe in oxygen. The same thing happens with a coral reef. All of these processes change the chemistry of the water that's lying over the reef. So as water moves from the ocean and across the reef, and of course water doesn't always move in a straight line across the reef. This is oversimplified. Water can swirl around and hang out on the reef for a long period of time. But as the water moves around on a reef and all of these processes are happening, the chemistry of that water changes, and we can measure the changes of the water chemistry, and that will tell us something about the metabolism and the health of the reef. And a previous study by our collaborator, Nancy Prouty, showed that when there was more influence of the submarine groundwater discharge at Kahekili, they saw more respiration and less photosynthesis and less calcification and more dissolution. So when there was less groundwater discharge, so less influence of that wastewater, the reef seemed healthier, it was growing more, producing more calcium carbonate and photosynthesizing more. And then when there was more of this groundwater discharge, it was dissolving more and respiring more and photosynthesizing less. So we are hypothesizing that we might see the opposite, that if we see an improvement in water quality, we might see the reef calcifying more and photosynthesizing more. Okay, so I'll give it back to Andrea.
So on March 26, our governor issued a mandatory 2-week visitor quarantine that dropped visitor arrivals to Hawaii by almost 98%. So we have almost 200,000 less people on the island per day. And as a result, the Lahaina Wastewater Reclamation Facility is receiving 40% less wastewater and injecting 60% less treated wastewater. So the big question is, how does this affect water quality and coral reef health at Kahekili? And additionally, there's been anecdotal reports of cleaner water at Kahekili and Kanapali and around the island. So this is a Google image of Kahekili Beach Park. It's right out in front of the Westin KOR. And we have 5 sampling sites, which are shown by these yellow pins. So the vent site where that groundwater is being discharged, that's located closest to shore, within about 5 or 10 meters from the coastline. And then we have multiple sites located around the vent. And then we have one site that's located down about 150 meters down the coastline. So these sites were chosen from a previous study with Nancy Prouty's work, and we're comparing our results to their data. So we're also sampling at the same sites. And these are some photos of our sites. So on the left is the seep site where the groundwater is being discharged. You can see it's really brown. There's no corals and there's high algae cover. The photo in the middle is a site that's a little bit further away from the seep site. You can see that there's still high algae cover, but now we're starting to see some color. We're starting to see some corals. And then the photo all the way on the right is our sampling site that's furthest away from the seep. So here, there's way less algae, higher coral cover. We're starting to see more diversity and more structurally complex ecosystem. So, you know, you don't even really need water chemistry to visually see the impact of the submarine groundwater discharge on this ecosystem. In addition to the kind of normal oceanographic parameters that we measure, temperature and salinity, we're also measuring dissolved inorganic nutrients, so nitrate, silicate, ammonia, and phosphate. And these nutrients are indicators of groundwater. So if we see higher concentrations of these nutrients, you know that there's more presence of groundwater, lower concentrations of these nutrients, and there's more influence of seawater because seawater has low nutrient concentrations.
And to look at acidification, we actually measure these two parameters, TA and DIC, and I won't go into detail about what that means. I'll just tell you that we can measure them very, very precisely. And then we can calculate all these other things that we're interested in. So we calculate pH, carbon dioxide, that carbonate ion concentration, and then this calcium carbonate saturation state tells us about how easy or difficult it is to make calcium carbonate. But the measurements we're making are TA and DIC.
Okay, so for emerging organic contaminants, again, two groups of different compounds. PFAS, we measure more than 30 compounds out of the 5,000 known entities in this class. So currently EPA routinely monitor 24. My lab now expand to about 33 compounds. And for personal care product, it's a suite of different compounds. Some example include ibuprofen, again, painkiller, naproxen, also another type of painkiller exists in brands like Reef. And these will be the indicators of wastewater discharge through household. But then we also have artificial sweetener and caffeine as a wastewater indicator that is through human usage. And also sunscreen, which is slightly different from the other PPCP because they are directly exposed in the water. So with all these compounds, we can actually understand what kind of chemical are the coral reef species are exposed to. So I love Katie's illustration about the metabolism, but even in human or marine metabolism, we often need to understand what kind of chemical stress that they're exposed to. So these two groups of compound will allow us to understand how this exposure will tie to the coral reef ecosystem. And I measure these compounds by using a magic box that is pretty expensive. It's called a mass spectrometer.
Okay, so we completed our first high resolution sampling effort in August. We're going to be doing another one in the winter, sometime in December or January. So we're collecting samples at each of those 5 sites 3 times a day, morning, noon, and evening, over 4 consecutive days. And the reason that we measure 3 times a day is so that we can catch that photosynthesis respiration cycle that Katie was talking about, or that light and dark cycle. And so the goal of the high resolution sampling effort is to characterize coral reef metabolism. When Katie and Yina and I first started talking about doing this work, we were really struggling to figure out how to kind of conduct such a large sampling effort when nobody was allowed to come to Maui. And so we were introduced to TOVA and to Huio Kovaiola, which is a citizen science coastal monitoring group on Maui. And they volunteered to help us with the field work. And so we really couldn't have done this very intensive sampling without their help. And I think it's just a really great example of how, you know, researchers, citizen scientists, and the community can all come together to conduct research that benefits our coastal ecosystems. So this figure, or this photo on the top right, is showing a Niskin. So we would drop the Niskin to the bottom, it would collect water, and then we deploy the CTD, and that measures temperature and salinity. And then we would kayak back to shore, collect the water from the Niskin, and kayak back out to our next site. And we did that 5 times, 3 times a day. So we are really appreciative to the groups and the people that helped us with the sampling. We're also doing low resolution sampling. So the purpose of this is to monitor for changes in water quality, specifically of the groundwater. We're collecting monthly or a little bit more samples at low tide at the seep site. It takes about 3 to 8 months for the water to get from the site of injection to the coastal seep. And so we're kind of keeping an eye out for those low concentrations, or that indicator that we're starting to see the low sewage volumes because of the population reduction. And once we see those low concentrations, we know it's time to get out there to do our next high resolution sampling effort to then characterize reef metabolism. So I'm going to show some very preliminary nutrient data. We just plotted this up within the past few days. So phosphate is on the y-axis, silicate is on the x-axis. Those blue symbols represent the data from our study, and the orange symbols represent the data that was collected in 2016 by Nancy Prouty and their group. And you can see that our data lie really nicely on their data. So that's a really good sign. I talked about this earlier, but high concentrations of nutrients, in this case phosphate and silicate, represent a higher concentration of groundwater in our sample, and then lower concentrations of these nutrients mean that there's more seawater in our sample. So the really nice linearity of this line tells us that most of that phosphate is coming from the groundwater. And this is showing nitrate on the y-axis and silicate. So again, those orange symbols are from the 2016 study, really nice linear line that's telling us that the nitrate is coming from the groundwater. And then this is our data. So we know we're collecting the groundwater because we're seeing high silicate concentrations, but our nitrate is very low. And this is puzzling. So we're not quite sure what's happening yet, but just to kind of throw a couple of possibilities out there, there could be some biological utilization of nitrate. So utilization in the groundwater itself, or bacteria utilization in the sediments before we're collecting that groundwater sample, or the groundwater sample at the seep sites. It could be also reduced nutrient loading. So we weren't expecting to see low concentrations of nutrients associated with lower sewage volumes quite yet, but maybe we're seeing it sooner than we expected. If that's the case, that would be fascinating, because that would mean that population has a large effect on the amount of nitrate that's released into the environment, and that we could already, that the changes in coral reef metabolism could already be happening in response to these lower nutrient concentrations, or the lower nitrate concentrations.
Okay, so another first look of the data, PFAS. So we detected one kind of PFAS, namely PFBS, without having to spell out the long chemical name. This is a short chain PFAS used since 2003 to replace a longer chain PFAS, PFOS, which has 8 carbon versus PFBS has 4 carbon. The 8 carbon variety was invented since 1949 and it was phased out since 2000. The reason being the government started to notice some toxic impact of this compound, and the reason to switch to a lower chain length compound is hypothetically they would decrease the bioaccumulation in organisms. However, dropping the chain length actually making them more water soluble. So that's why now we're seeing PFBS everywhere. So the good news is for Hawaii, for this sampling of the more than 30 PFAS, only 1 compound detected with variable concentrations. And also just to have a contrast on the left-hand side, this is the Galveston Bay concentration that we observe for PFBS. And in Galveston Bay, we often observe more than 1 PFAS compounds, and the PFBS concentration vary substantially. And in March 2019, there's a substantial concentration is actually coming from the ITC fire where a bunch of firefighting foam was applied. So compared to Galveston Bay, which is a drainage basin collecting very large scale of metropolitan area from Houston and Dallas watershed, I think the water is really clean, and I'm very envy you guys for the great water quality. However, it's nonetheless also alarming to be able to even detect trace amount of PFAS in seawater. This means somehow some sources are being transported to the coastal water, and even at low concentration, there's nothing alarming in terms of human health, given the vulnerability of the reef system. I think this is still worth monitoring, especially if we can do a longer term study to understand the variability of these compounds and see if other PFAS may show up as tourism increased with other applications. So this is a first look. My lab is still trying to extract all the other water samples for the pharmaceutical and personal care products. So it's really exciting to see this, and very exciting journey.
And you guys will have to hold on to see acidification and metabolism results because my lab is behind on analyses. But Yina and Andrea were able to get out some early results. So I'm really happy that they had some data to show you. So I'm just going to summarize for you. So Hawaii's had this astonishing decline in tourism that apparently only a global pandemic could bring about, which, while horrifying, is providing this amazing scientific experiment that we could have never designed on our own. And this is providing a dramatic decline in wastewater injection that is ending up in the submarine groundwater discharge that ends up in Kahekili. So our project is to look at the impact of this wastewater decline on 3 different types of water quality: nutrients, emerging organic contaminants, and acidification. And then to connect these changes in water quality with a measure of ecosystem level coral reef health, which is coral reef metabolism. And this is really exciting for a couple different reasons. First, to my knowledge, no one has ever tried to even look at the connection between emerging organic contaminants and coral reef metabolism. As Yina's explained, there's a lot of reasons to think that organic contaminants could affect coral reef metabolism, but I don't think that anyone has ever even looked for a connection between these things. So this is sort of a first look at how these might be connected. And second, The studies that have been done so far looking at the connection between nutrients and acidification on coral reef metabolism have been in the negative direction, including my and Andrea's work, looking at negative impacts of increasing nutrients and increasing acidification on coral reef metabolism. And this is an unexpected and sort of incredible opportunity to look at potentially the opposite effect. So we're very excited to see if we see any positive impacts of changing water quality on coral reef metabolism at Kahekili.
And that wraps it up. So there's a lot of people that, you know, have been contributing to the field work and contributing their knowledge to this research. So we just wanted to thank everyone, and thanks again for sitting through our talk. And I guess I'll hand it back to Liz.